would like to thank BCSIR for accepting my abstract and giving me the opportunity to present my research here. Honorable Chair, my direct teacher, co-chair, and this presentation is a part of my postdoc research which has been carried out at King Fahd University of Petroleum and Minerals, Kingdom of Saudi Arabia. I would like to talk on recent advances in the adsorptive removal of tetracycline from aqueous solution. Now I would like to talk a brief background of my research. In the aquatic environment have caused worldwide problem. In recent years, tetracycline is a widely used antibiotic because of its effectiveness and stable structure. About 70 to 90% of tetracycline can be metabolized by the organism and therefore ends up in the soil, sediment and aquatic environment. It enters the aquatic environment through pharmaceutical wastewater, medical waste, veterinary waste. So it is very essential to remove this contaminant from wastewater. Now the sources of tetracycline in the environment. Here I have shown the various sources from where tetracycline enters into the wastewater and then contamination by tetracycline antibiotic. Now the nature of tetracycline. Here we can see tetracycline is an amphoterary compound with a unique structure comprising 4-ring system, and it can exist 3 different forms depending on solution pH, and it has got 3 acid dissociation constant. And adsorption property of this tetracycline mainly depends on these 3 different forms such as cationic form, anionic form and deuterionic form. Now, how we can remove this tetracycline from wastewater? Well, several approaches have been employed. For example, adsorption, degradation such as photocatalysis, photochemistry, electrochemistry and other methods. However, adsorption is a universal method and it is very simple and easy, and it does not produce secondary sludge which can further pollute the environment. So our approach is adsorption process. Now, how we can remove this tetracycline? Well, various types of adsorbents can be used for the removal of tetracycline from water system. For example, carbonaceous materials, metal and metal oxide based composite, metal organic frameworks and other adsorbents. Now I will show some typical examples of the application of various adsorbents for the effective removal of tetracycline. Here is the carbonaceous adsorbents. So these are the typical examples for the application of various carbonaceous materials for the removal of tetracycline. As we can see here, that the adsorption capacity of tetracycline depends on the nature of adsorbents as well as the experimental conditions. And it is interesting that in most cases, pseudo-second order kinetic models as well as Langmuir models specificated the adsorption data. Then comes BCSIR. Here are the typical examples of the application of BCSIR for the effective removal of tetracycline. Then I have shown metal oxide and composites. These are the typical examples for the application of metal oxide and composite for the effective removal of tetracycline. Then zeolites. Then clay or phyllosilicates adsorbents. Then polymers or biopolymers. These are the typical examples of polymers and biopolymers. And then miscellaneous adsorbents for the removal of tetracyclines. Now, the interesting part is how this tetracycline bind to the adsorbents. Here I have shown typical example. So tetracycline can bind to the adsorbent by pi stacking, pore filling, electrostatic attraction, and hydrogen bonding. Now, how can we propose this mechanism? We have three strategies. Number one is adsorption kinetic and isotherm modeling. Here I have shown typical example of adsorption kinetic and isotherm modeling. And these models give information whether tetracycline form monolayer or multi-layer onto homogeneous or heterogeneous surface. Our second strategy is spectroscopic mechanism. Spectroscopic studies let you know how tetracycline binds to the surface in aquatic system. Here I have shown the typical example of FTIR spectra of BCSIR before and after tetracycline adsorption. Finally comes surface complexation modeling. It let you know whether tetracycline form inner sphere or outer sphere complexes during removal process. So outer sphere means there must involve a water molecule between tetracycline and surface. On the other hand, in case of inner sphere complexation, tetracycline will be bind to the surface functional group directly. That means there is no involvement of water molecules. Then challenges and future perspectives. More innovative research on the toxicity of spent adsorbents in aquatic environment is needed, and only limited study has been used for surface complexation model to predict the mechanism of tetracycline, and artificial neural networks, machine learning and surface response methodology can be used to gain new insight on the tetracycline adsorption process, and competitive adsorption of tetracycline with other pollutants requires special attention because remediation is often performed in the natural environment, and more research is needed to understand how this contaminant is bind to the surface in natural environment. So these are the conclusions. This study summarizes previous work on the uptake of tetracycline using a wide range of adsorbents from water and their adsorption mechanism. And it is interesting to mention here that the removal efficiency of tetracycline onto adsorbent mainly depends on several factors, for example, nature of adsorbents as well as experimental conditions. And finally, more research is needed to study mechanism of adsorption using novel adsorbents. These are the references where from most of the information have been gathered. And I would like to acknowledge King Fahd University of Petroleum and Minerals for providing me a postdoc opportunity, and Ministry of Higher Education, Government of Saudi Arabia for giving me full funding for postdoc for 2 years. And I am also grateful to Aminul University of Science and Technology, where I am Associate Professor of Chemistry, for granting me study leave. And finally, thank you all for this session. These are the King Fahd University campus.
Thank you, Dr. Aminul Islam. Professor Adsorption. Now the floor is open for questions.
So I have two questions, please.
Sure.
Yes, Professor. So my first question is, your research is going on only tetracycline, or do you have any research or any information regarding other antibiotics like amoxicillin or tetracycline?
We are studying tetracycline, paracetamol, and pharmaceuticals
My second
using various innovative adsorbents. And recently we have published 6 papers in high impact factor journals. I have not shown here, and more research is going on.
So I need one information. So you are carried out on wastewater. What about the other parameters like wheat, paddy, rice, or chicken?
We can consider this in future, but our main target now is to deal with wastewater.
So what is your solution? We should take an initiative regarding this issue.
Yeah, it can be taken.
This is a
Yeah, it can be taken to collect wastewater from various sources.
What about food chain? When I tested our blood, the whole doctor or whole pathologist said your body is resistant from this type of antibiotics. That's why I'm asking.
Yeah, nowadays tetracycline, special antibiotics are major problem, as I said. So you can consider this.
Okay. Thank you.
Actually, his main work, the speciation of the tetracycline, and he can make the selective way
Yes, I
for
understand.
the removal of the tetracycline as well as other adsorptive.
I just want to know that any other researcher doing this type of job in Bangladesh or in world?
Yes,
You
some
know-
other groups are
you know
doing
Bangladesh?
some groundbreaking research work, but ours is different because we are using
This we need.
yeah
conduct
because
this
we are
type of
using
research.
biomass-based innovative adsorbents, which is not being used before.
Thank you.
Actually, this is model work, but I'd like to know the half-life of the tetracycline about 10 hours, for example. So when the tetracycline come from the factories, the tetracycline plant, the antibiotic plant, is by the wastewater to the aquatic system. So if the half-life is 10 hours, so how the presence of the tetracycline make harmful to the aquatic life as well as human? The
Okay.
connection.
Okay, thank you so much. As I said, that tetracycline remains 3 main forms. So if half-life passes, it can change from one form to another form. These 3 forms are harmful. It can change the structure of the tetracycline in the natural wastewater state.
No, the half-life means it must.
No, it is stable molecule. Tetracycline itself is a stable molecule. It is not easily break down to other molecules. It can change from one form to another. That means if solution pH is low, then it can exist as cationic form. On the other hand, if solution pH is neutral, then it can exist either positive or negative cationic,
No,
deuterionic form.
PK1,
PK1, yes.
3 point something.
Yeah.
That means if we maintain the pH of the solution more than 3.5, that must be anionic form.
No,
PK1.
3.3 to 7 point something, it will exist deuterionic form.
Deuterion never be a range.
Yeah.
Deuterion should be very unique, very narrow range of the pH. Because already you mentioned 3 pK value,
Yes.
pK1, pK2, pK3. So it might have 3 acidic proton, okay,
Yeah.
acidic hydrogen. So first pK1 means 3.3. If we maintain the pH more than that, so the predominant species should be single deprotonated species. If the pK2 like 7 point something or 7, if we maintain the pH more than that pH, so the predominant species should be deprotonated species, dianionic. The first one is monoanionic, then dianionic. So if the very stable, so we never take more tetracycline. One is enough. So the antibiotics we usually take after a fixed time, like either 8 hours or 12 hours or 24 hours. That depends on the half-life of the antibiotics. That I know. Maybe I might be wrong, but why we take the antibiotics at a fixed time? Because if you take the antibiotics now, so within, for example, if the half-life is 8, so after 8 hours, the level of the antibiotic in our body, that should not be prevent the growing of the bacteria. That is why after 8 hours we have to take the second antibiotic to prevent the growth of the bacteria. This is the mechanism of the antibiotics.
Okay, thank you so much for your suggestion.
Thank you.
Thank you.
Thank you all.